Hello friends, welcome back to my channel Xin and Protein X. In this tutorial video I will discuss about the very basic and important question that why does Dpn1 restriction enzyme cleave only parent strand but not the amplified strand. Many of us we use Dpn1 for the parent strand digestion after site-directed mutagenesis or any kind of mutation. So we normally use this kind of enzyme. But the question is, why this kind of Dpn1 restriction enzyme, why only cleave the parent strand but not the amplified strand? So to answer this question, I have to go little deeper about the theory. So I drawn one of the picture, that is there is schematic diagram of the restriction enzyme, how it works. So here you see the bacterial DNA, the bacteria and the bacteriophage. We know that bacteria it attacked by the bacteriophage DNA, and then normally the bacteriophage DNA they incorporate their DNA into the bacterial genome, and so after that, so bacterial DNA when they amplify the bacteriophage DNA, so when the lot of bacteriophage DNA inside their bacteria, then they destroyed by their DNA, by the foreign DNA. So to protect this, they utilizes one of the enzyme, that is the restriction enzyme. So they utilizes this restriction enzyme to protect from the bacteriophage attack. So how they do? So they actually in the bacteria they produces lot of restriction enzyme, and this kind of restriction enzyme they find some specific sequence and they cleave that DNA, that sequence. So for example, there are in the E. coli system they have the E. coli, one of the restriction enzyme, and this restriction enzyme it find this kind of sequence. This kind of sequence, and they cleave this sequence. So whenever they find this kind of sequence, they just recognize it and they cleave it. So now question is that this kind of sequence it may also present in the bacterial DNA, right? So the restriction enzyme, their job is just only recognize and cleave. So whatever the DNA it is, foreign DNA or own DNA, they will recognize and cleave it. So if it happens, so also the bacterial DNA also be susceptible by this process, right? So that's what they have their own system. What they do, they have the methylase. They have the methylase enzyme. They have the methylase enzyme, and the methylase enzyme, this kind of enzyme, their job is to methylate to add the methyl group to their own DNA. So they add their own sequence, the methyl group, and this is how restriction enzyme distinguish the foreign DNA and the own DNA. So in the foreign DNA there is no methylation, but in their own DNA they have the methylation. So restriction enzyme now they distinguish their own DNA because that is the methylated, and they won't cut or won't cleave their own DNA. So they only recognize the foreign DNA sequence and they cleave it. So this is their job. This is how it works. So now I'll go to the next part, that how Dpn1 then recognize this kind of sequence. So for that I have to go to the next slide. So let me erase this first. So here you see there are two different, you see this is actually the PCR reaction. So when you start any PCR reaction, so there are most of the product are amplified, but there are very little concentration of the parent strand it exists in the PCR product, so that not be amplified. So basically what the template that we use, like plasmid or any kind of DNA product that we isolate from the bacteria, the plasmid. For example, we use here the plasmid. So this plasmid we isolated from bacteria. So this kind of strand, you see the parent strand, it is not amplified. So this template contain this methylation because this is isolated from the bacteria. But in the amplified product, there is no this kind of methylation because this is the PCR amplified product. So that's what Dpn1 is one of the type 2 restriction enzyme, and they recognize this kind of sequence GATC, and they recognize the GATC, but they cleave only if this is adenylated in the adenine portion, if there is any methyl group attached. So that is methylated adenyl. So if this kind of sequence is there, if any this kind of methylation is there, then Dpn1 recognize and cleave and destroy this sequence. So now I hope that you almost got your answer, because Dpn1 it may recognize this kind of sequence here also, it may present here also in your amplified product, right? But here there is no methylation, because this is the amplified product. But here this kind of sequence is there also, and also there is methylation because this is from the plasmid that is isolated from the bacteria. So this is why Dpn1 recognize this sequence and cleave it and destroy this sequence. So after that you will get only the amplified product, and in this amplified product no parent strand present. So I hope you understand this part, and this question answer is very clear to you. And if you have any queries about it, you can write in the comment section. If you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks guys.